Okay. Sorry about the always awkward beginnings. It's just the trying to get everybody admitted and trying to get documents up and all that. It does my old head in, I'm afraid. Okay. So quiz is over. First hurdle done. To be honest with you guys, like looking at the responses in the chat box, it's pretty much what I would have expected. So there's some people that are quite happy, some people that got a bit thrown off, some people that ran out of time, some people that were, you know, what didn't quite go the way that they wanted it to go. And that's really normal, but part of it is, you know, once you've done it, now you know exactly what it's all about. So the next one will be the same thing, same format, same everything, and you've kind of broken its back, for want of a better word. Yeah. Do you at least feel like relieved that it's done?
Yes.
Yeah. Good. Yeah, so it's just important to also give yourself a bit of a break too. Like, it is the first quiz in this subject. So of course you're not going to, you might have done it perfectly, but it would be weird. You know, like it's okay not to have a perfect handle on absolutely everything, but we are going to talk about that as part of this tutorial as well. So without further ado, where's my thingy? Okay, there it is. Okay, so one of the first things, oh God, hang on, there's people in the waiting room still. Yep. Okay. I swear, if it's more than 5 minutes late, I'm just going to be mean and nightery and say you have to wait outside. Okay. So cells and tissues is where we're up to. But before we go any further, oh God. Sorry, waiting room. I'm going to fix that setting just so that everyone's automatically admitted, because it drives me a bit spare. Okay. One of the things to take into account with all of this is really these dudes. Okay, so at the beginning of every single part at each topic, so you have part 1 and part 2 and part 3, you will see that there are a bunch of learning objectives, and they're not just there for fun. So all of your exam questions are written directly from learning objectives. So as long as you can tell yourself that you know all of these things, then nothing will surprise you in the exam. We can't ask you questions that aren't connected to these learning objectives really, really tightly. Okay, so what I would suggest, when you've done a topic, and particularly using this for sort of revision purposes, go through all the learning objectives that you're supposed to have understood, and highlight the ones you know and highlight the ones you don't know, and pay particular attention to those ones. Yeah, does that make sense? Because you will, I guarantee you, get questions out of all of these, and these are what you're supposed to take away from these topics. Okay, and I actually am going to have to put yet another document into the chat, because that's the way it goes. Just a second, guys. But it's only 2 so far, so that's something. Okay. Alrighty, so having a look at that, that's the things that you're supposed to have taken out of your lectures and readings and all the clips and things you've got on Interact 2 for this week. So you can guarantee that in the next test, all of your questions will be based on this stuff. Okay, and remember we talked about having a really good look at what's on this and going, what's going to be a multiple choice and what's going to be a short answer. And it's really easy to actually figure it out, because if you've got describe kind of questions, of course they're going to be the ones that the short answer questions are based on, right? That's the longest response you're required to give, isn't it? So, you know, have a look at the ones that say describe. So 5 factors affecting the rate of diffusion would be something you need to go with. Different cell shapes. Why do we have different cell shapes? What's the structure of epithelial tissue? How does three examples of how structure and function are related in epithelial tissue. How is the anatomy of connective tissue related to its function? All of those are, one of those will be your short answers. Of course it will, right? If not a couple. Things like list and define, often they're going to end up being in your multiple choices more often than not. Okay, so just be careful and grab them. Really, really important. Okay, having said that, let's see what we do know, because we're going to go through our usual tutorial document and see where you're sitting. So is someone trying to talk, or is that just a badly muted person? All good? Yes, it's just the not mute button. Okay, alrighty, have we all got that document downloaded, working, etc.? Do I need to put it up again? Yeah, Catherine?
Can you hear me? Yeah.
Yep.
I haven't had anything come through in chat. Just Andrea has said that they can't see it in the documents in chat either. So that's the only thing I've had come through.
Okay.
I haven't seen the document. I'm sorry.
Let me try it again. Okay. Here we go. This is third send. Let's make it work. Come on. Go through. Make sure everybody's got it. Yes?
Yeah.
Jolly. Okay, excellent. Alrighty, and don't forget, all of these documents do actually go up on i2 like the week after you've done the tutorial. So if you had to, and it's not ideal, but you can go in and get them in retrospect as well. Okay, moving right along. I can hear small people. This reminds me of myself studying. I had to study with a toddler and it was always interesting. Okay, multiple choice questions. Don't forget, this is just to check on where you're at. So see how you go. All right, let's go. All right. Number one, a needle piercing through the plasma membrane, would it travel through what? Have a look and then we'll find some answers. All right, what do we reckon?
Definitely C.
Definitely C. Votes for C?
See, yep.
Yep. All happy with that? Good, because obviously just keep telling phospholipid bilayer, which you've heard a lot in the last week, I'm sure. Bilayer, 2 layers of phospholipids. Very good, excellent. The cell's energy comes from what?
B.
B.
Yep.
B. Everyone fine with that?
Yep.
Yep, good. Cilia, interesting. Okay, maybe if they wiggled fast enough, I don't know. All right, now this is important because people for some reason get quite confused between diffusion and osmosis. So someone's sitting at a bar smoking, bad people, that's terrible. By the way, I still smoke, it's terrible. All right, so I'm trying to quit, but you know, said every smoker ever. So someone's sitting at the end of the bar smoking, annoying you, and it's a bar that actually allows smoking, which couldn't happen. What process is involved in the movement of smoke away from its source?
A.
A?
Yeah, diffusion.
Yep, definitely not B. Why? Someone tell me why.
Because osmosis involves water.
It does. It does. And it's also at the opposite level of concentration as well. Facilitated diffusion. Well, you know, I guess that could technically be an answer, sort of, except that nothing's really facilitating except for air. It's not exactly using energy to get there, is it? Not that we know of anyway. In current physics, we don't know. And endocytosis, we're not removing anything from a cell. It's literally bar smoke. Okay, here's a question for you that's not on your question paper. What diffuses through air faster? Fine French perfume or fart?
Fart.
Actually, the answer is fine French perfume. So technically, if I sprayed myself with perfume at one end of a room and also farted at the same time, this is just a lovely image for you to take in. The perfume would reach you faster
Is it
than the
because it's a
fart.
stronger thing?
Yeah, it's actually because of the molecule size.
Yeah.
So actually talking about the size and the fineness and the density of molecules. So believe it or not, a fart is heavier.
Depends on the fart, I guess. If it was my partner, you would smell it first.
Yeah, possibly. Well, it depends on whether you use the French perfume. And then also there's guys that just are brilliant at that sort of stuff. And it's like literally a talent that they, practice, I believe.
I'm totally going to use it at the dinner table tonight.
Right? It's really interesting. And well, actually you could test it. See what happens. Bring your perfume bottle
I
down.
won't be testing that.
I don't know, little toot, and then say, so which one can you guys smell? That'd be fun. That'd be just a non-mum thing to do, right? Mom! We're all crazy somewhere. Okay, move on. Which of the 4 tissue types are the ones that cover or line the organs?
D.
D.
Yep. Epithelial covering. Yep. Good. And tendons and ligaments are what kind of connective tissue? Oh,
You just answered.
You said connect.
I think I might be tired. It's possible.
B.
It could be B. Yeah. Don't you wish you had me in the room when you're doing the test? Because I'd give it away if I was tired. Anyway, yes, it is B. Good. Honestly, though, were you going to get that before I just had my little verbal vomit? Yeah. Okay, good. Sorry. It's been a day. It also was supposed to be, the weather said today that it was going to be nice and autumnal. I was really looking forward to it. And then I went to work in Bathurst, on Bathurst campus, and it was like 33 degrees. So not happy, Jan. I'm just too tired. I don't do heat well. I tend to wilt like a plant. I'm not good. Nice and cool and I'm happier. Okay, now, we know this fella. We've seen him around. There's quite a bit on this today. So just giving you a quick look at our old mate, the prokaryotic or eukaryotic cell.
Eukaryotic.
Yes, absolutely. Because if it was prokaryotic, it wouldn't have all of those bits and pieces, and also we'd be talking about bacteria. We're not. We're talking about human cells at the moment. So lots of different areas, and the thing we're talking about at the moment is the cell wall. So I want you to take a couple of seconds and define passive or active transport. Remember, not a novel, just define. So tell me specifically what is the difference between passive and active transport across a cell wall. Just take a couple of minutes, consult your notes, put together a few sentences, the sentences that you would put together for a test, and then we will ask some people to share what they've got. So just take a couple of minutes and see if we can do a nice, simple, say 2 or 3 sentences on the difference between passive and active transport. So if you can, if you're in a place where you could just jump in and speak it right now, then just be happy about that. That's really good. Right? But it also might be something you need to look up. Oh, actually, while you're going, who did the test sort of from their brain and didn't use notes a whole lot? Did everybody use their notes at some point?
Yes.
Yeah.
A little bit.
A little bit. What about multiple choices? Were you able to just kind of whack through those pretty quickly?
Yeah.
Good.
Yeah, those
Yeah.
were, yeah.
Good. Yeah. So it was only really the short answer stuff that you had to go consulting for. Yeah.
Yeah,
Did anyone run out of time in the test? No?
Yes, I did.
Yeah, I did.
You did?
I did.
I
Okay.
got
Questions.
I had to rush
Yeah.
my last few questions because of my time. Would have liked to have answered more deeply, but I couldn't.
Okay, so that's a really, really good heads up. We've been talking about it the last couple of weeks, but it is a good heads up for exactly how fast time does go and how much time you really can't spend agonizing over multiple choices. You've got to get through and get to the next answer, the next question.
I just wanted to say, be aware, guys, the questions go from simple to most complicated, so save time for the last questions.
Yes, and or flip it. And because you can actually just scroll backwards and forwards throughout the questions. So another strategy might be to start at the end. Like actually start at the harder questions, get those out of the way, and then go back and work through the multiple choice if you want to.
I was gonna do that, but it says the moment you click next, it saves the question and you can't go back.
Oh, but only if you don't answer it.
Okay.
So yeah, the other thing you'll find is at the end, before when you hit submit, it will ask you, it'll actually say, oh, it looks like you've missed this question, and this question. Do you want to go back and do them? And then you just hit OK and you can go back and do them. So there's a number of fail safes built in. All righty. Now let's see some brave people. Maybe tell me in your words, nice basic English, not too much jargon. What's the difference between passive and active transport in a cell? Yeah, you go, Alison.
Jump in.
Go. Yep.
So passive transport doesn't actually require any energy to move the cells. So like moving down a gradient or something
Yes.
like that.
Yes, perfect. Good. Anyone else got something else in their words? That's perfect, by the way, Alison. Great.
Active transport is when the energy is used to move against the gradient.
Yes, absolutely. And for the high distinction, what's the energy molecule that we're talking about there?
ATP.
Yes. Put that into answers, guys. It's always that, active transport uses ATP and passive transport does not. It's a big one. Yeah, really, really important. Anyone else got something to contribute on that one? You're very quiet today. Okay. All right. That's because I'm going to give you the answer. And I am. Okay. If I were you, I like little tables like this, little comparative tables that just have one on one side and one on the other. Okay. So no ATP versus ATP, down a concentration gradient, as Alison said, as opposed to against a concentration gradient. Depending, we could be using protein carriers or transport molecules in passive transport, but we definitely have to use them in active. And this is examples as well. So osmosis is passive. Simple diffusion is passive. Endocytosis and moving large molecules into a cell, that would be active. Yeah? Just if we can try and hit all or some of those points, that would be kind of the HD level answer. Yep. Energy, concentration gradients, what's going on with transports and or carriers, and give me an example. Yep. Not that I'm marking your tests, but give us an example, I should say. All righty. Let's move on to our next piece of excitement. Where's my thingy gone? Oh, there it is. Okay, this is easy. This is good, right? This is our picture of a cell. All you got to do is tell me what stuff is being pointed at. So yay, right? This is a good one. Top left-hand side, what are we pointing at there?
Golgi. Or Golgi.
A Golgi complex?
Golgi, yep.
Yeah. All right. What about the one underneath it?
Oh no. I thought that was the mitochondria.
It is the mitochondria. I don't quite know what's going on there. Okay, anyway, never mind. The top right-hand side?
I remember if that's
Smoothie.
Smooth. ER.
Rough endoplasmic reticulum.
Yes, you know what I think? I think that one, the mitochondria, the arrows in the wrong place, was supposed to be pointing at a Golgi. Yep, okay, that's what it was. And of course, the last one, if you don't get that, I'm seriously concerned. Nucleus. Yeah, it would be a worry if you'd gotten through the full week and you were not quite sure where a nucleus was. That would be upsetting. Okay, good. I have a funny feeling that one of those arrows has been misplaced. I do apologize. I pulled this directly off Nat's work, so I'm going to tell her tomorrow. Naughty, she put things in the wrong place. Okay, that one was nice and easy. Alrighty. Now, descriptions. So again, no explanations, just descriptions. So again, I'm going to give you a couple of minutes to pop together some kind of answer for this one. And this is where lecture notes do come in handy, because it says the appropriate level of detail, and that's always quite hard to figure out, but that's why you're given the lectures and the lecture notes, is so that you know the level of detail. So have a look through, see the level of detail that you've been given on this, and then pretty much spit it out in your own words.
How do you say that? Squamous? Squamous?
Close, yes. Squamous is general, but you know, if you're American, you'd say squamous. So you could, all you have to do is just have an accent for a second. It's fine. Yeah. So next time somebody says you're mispronouncing that, say, no, I just spent like 6 months in the States. It just comes out every now and then.
Squamous.
Yes. Squamous is one. Yeah. What's another one?
Discoid and fibrous.
Yes, absolutely. Good. Everyone agree? Yep. Perfect. Perfect. And that is the level of detail, isn't it? That's exactly what you were given, just the slide that I've got next, which is literally, this is what you were given, right? Squamous, discoid, and fibrous. And again, if I were you, I would say, so you've been asked to describe what you see. This is pretty much what you'd want to write. So not just write squamous, but tell me that they're thin and flat. If you remember that they've got a bulge at the nucleus, yeah, but other than that, thin and flat would be fine. Discoid, disc-shaped, duh, and fibrous, long, slender, thread-like, etc. If you can remember an example, that's a fully HD question. Yep. Okay. So pretty simple, that one. Different
Can I just ask a
cells.
question?
Yes, of course.
So if we have to put that in our own words, like do we have to? I mean, like, it's,
Well, yes.
how do you,
So you
describe
well,
disc-shaped apart from being disc-shaped?
you might need to write shaped like a disc.
Okay, well, that's
Yeah.
fair enough. Yeah.
Yeah, like you just need to sort of, because you can't rote memorize lecture slides, because technically it is plagiarism. So shaped like a disc appears disc-like in shape. You know, however you can rearrange it, that would be good. But yes.
Okay, yeah, that makes sense.
Yeah. Just don't rote learn lecture slides, because we have had that problem before, and it is tricky. Okay. So now you're going to explain. So what we're looking at explaining is the selectively permeable membrane. And again, always imagine that you're explaining it to, you know, like a 10-year-old or a 12-year-old. Keep it simple in your head, but anticipate the whys. Right. I think I said this last week, like my child is always asking me why every 2 seconds. There's no such thing as a proper explanation. It's just always why is the next answer. So, you know, you could talk about your selectively permeable membrane. What does it select for? Why is it selectively permeable? What's the difference between that and non-permeable? What's allowed in and what isn't allowed in? And why? So a couple of minutes, and then I'll get you to other people, please, than just the usual suspects. So Emily, you have to shut up. Angela, you have to shut up. Because I want some of the quieter ones to take a bit of a risk and say something. It'll help you. It'll help you to verbalize what you know, or even what you don't know. Pardon me. Okay, does anybody want to give that a whirl? What is met? See, this is, you should answer questions early in the tute, because it
I'll
gets
have a go.
Thank you.
All right. There's a bit that I want to, the end part, I'm not quite sure on. So I've written that it only allows certain substances to pass through the cell, and it does not allow anything through that is large or charged.
Yep, absolutely. That's really, really good. Maybe it would be better to say allow through the membrane rather than through the cell.
I had membrane sitting there actually.
Yeah. Just because it's action or movement of stuff through a cell is slightly different than movement across a membrane. And we are specifically talking about the membrane. So yeah, but other than that, that's pretty good. So okay, let's add to that, guys. What else have we got?
Is it there's 3 ways that it can be selected? Like it can be transported in 3 ways. It can be like put into a balloon.
Yep.
And then one can be like a gradient, like using electricity
Yep.
charges.
Yep.
And then the other one is through passive, which is like, is that it?
Yeah, absolutely. Nice. Really, really good. Actually, you can put those 2 answers together, and then we've got a really nice kind of broad answer. I really like that. Has anyone else got anything else to add that they think should be in there?
Add to that. They only allow certain substances in to maintain, sorry, what was I trying to say?
The theme, the stability,
to maintain an internal temperature or environment.
Yes, absolutely. Which we're going to talk about in some detail shortly, actually, in terms of what happens if things come in and go out when they shouldn't. But yes, put all those things in together. That's great. So you had mechanisms there with dye. You had all of the different examples. Give me examples of what is movement going through, and then Chloe, why? To maintain the environment of the cell. Yep.
Catherine, would you say that it is, I'm not sure if I've got it the wrong way around, but water can come and go through the membrane, but solutes can't go into the membrane, but they can leave the membrane?
Yes.
Like come out through the membrane. Is that right?
Yes, absolutely.
Yeah.
Water does come and go, and that's one of our biggest problems actually in nursing, is actually the movement of water in and out of cells. It's one of the trickiest things we've got to manage, which is why we're going to jump on to the next thing and talk about our old mate osmosis, which is pretty important. Oh no, I've just given you a thing there. Okay. Certain substances controls what enters and leaves the cell. And as you can see there, a number of things will go through and a number of things will not. So anything that's bouncing off obviously is a not. Polar organic molecules, sugars, most ions require transport, and obviously large proteins. Certain things can diffuse easily though, of course, and that would be water, oxygen, and carbon dioxide, because they do need to do that. All right, that would be the combination of the answers that you just gave there is lovely. I would be very, very happy with that. All right. Where am I? There. Okay, now this is a bit chemistry. Okay, so it's a thought experiment, and it is tricky to do. Now, I'm annoyed about this one because I'd love to do this one with you guys actually in situ. So the way that we would normally do this is we would get ourselves a cell, and what that generally is an egg that's actually had all the calcium carbonate taken from it with acid, and it just lives us with the plasma membrane literally in a big fat nucleus in the middle, which is great. And then we put it into a solution and see what happens to the egg. But here we're talking about different concentrations. All right, so we've got to look at these beakers and figure out which ones are which and the technical names for them. So if we have 90% water and 10% solute, let's just assume that the solute is salt for the moment. Okay, so it's literally, we're talking about like saline. So 90% water and 10% solute is what in terms of, you've got hypotonic, hypertonic, and isotonic solutions.
Is it hypertonic?
Other way around. Hypo, yeah, because it's under. So there's a little amount of solute and a big amount of water. Yeah. What about the one on the I should have started on the left. The one on the top left, 63% water and 37% solute.
Hypo?
Hypo, do you think? Which one looks most like what you would think human blood would look like? This is going back to you, how much blood do we have and how much solute? And we did the components of blood and how much is in what? Okay, so that one would be, so top left would be your hypertonic, right? 37% solute, yeah? And then we've got hypo over there. What do we have if we've got the bottom left-hand side? Which one's isotonic? Which one do you think is closest to normal blood?
%75%.
Yep. Yeah, exactly. So which makes the next one what? Slightly hypotonic. So you've got one that's very hypotonic, but that one would be slightly hypotonic. So we're just going to see less movement, basically. So if we have a hypertonic solution, there's more solute outside the cell than there is in. So outside is super salty, and the inside is just a bit salty. Which way is the water going to go?
Out of the cell.
Out of the cell.
Absolutely.
Absolutely. Yeah, because it's water's job in this mission, and its mission in this life is to dilute the everythings. Apparently, that's what it does. So it goes against concentration gradients, and it goes, well, it's way so salty out there, I have to go dilute it. Oh, bugger, I just killed the cell. Oh, okay, never mind. And the opposite, so hypotonic solution, on the other hand, if I've got all my solutes inside the cell and nothing outside or very little outside, where's water going to go?
Into the cell.
Into the cell. Yep, that's right. And literally, what's the scientific word for explode?
Lice?
Lice, yes, that's right. Lice, cell lysis. And of course, if we have an isotonic solution, this is what we give patients all the time, right? This is the whole point of giving them normal saline, okay? So that we don't explode or crenate their cells. I should say lyse or crenate those cells. Super important. If you want to do this experiment, by the way, it's heaps fun. Get an egg, soak it in vinegar for a few days, get rid of the actual shell, the calcium in the shell. It leaves you with a membrane. Put it in really salty or sugary water and see what happens to the egg. And you've got to keep weighing it. So it's a very little science experiment. So the egg's going to lose weight, which is lots of fun. Wouldn't be great if we worked for us as well. Like I'd maybe just go jump in a salty bath and all my water will get drawn out and I'll lose a few kilos. Yay. And then pop it into something opposite. So pop it into like distilled water and see what happens and watch your egg grow. It's very exciting. Okay, is that clear to everybody, or would you like, I can try and explain it differently if it's not clear. Water will go wherever the greatest concentration is. And you've just got to remember the words hyper, hypo, and isotonic. Yeah? Okay.
Could we just run through them again? Because I missed that there was the two figures in each of the little tubes.
Yes, of course. So do you want to actually go through the figures or the concepts?
Probably the figures.
The figures, okay. All right. So our first top left, where we've got, I don't know, so we've got 63% water and 37% solute there. And at this particular point, that's what kind of compound did we decide that was? Hyper or hypo?
Hypo.
Hyper, yes, exactly. So this is a hypertonic solution. So what is going to happen, as you can see, right? Our egg's got 90% water and outside's only got 63% water. Water's job is going to go out there and give it more. right? It wants to dilute. So the water is going to leave the egg. So if you're going to draw arrows, you'd be drawing arrows going out. Yeah. Okay. If we jump to the opposite and we go into a hypotonic solution, when we're going down the bottom, the opposite thing is going to happen. Okay, so we've got more water is going to be coming in to the egg to try and dilute. Right? If we have isotonic, there is even movement, so you can have arrows going out and coming in. And it is true, guys, it's not like water only goes in one direction. It actually does go in both directions for all of the above, just in greater amounts. Okay. In an isotonic solution, the water is going in and out at an even rate, okay, going through the selectively permeable membrane. And that's what we want in general in a hospital setting. Okay.
So there isn't an isotonic example on the screen right now, is there?
Yes, top right.
Top
See,
right.
Yep, because
Oh,
you've
yeah,
got same water, same solute. Yep. So that would be completely even, no net movement either way. So you need to look at the amount of solute and the amount of water in comparison to what's going on inside that egg.
Catherine, sorry,
Yep.
on the worksheet, there are two more at the very top that I don't think, yeah. So the first one is 50% water, 50% solute in the cell, and 50% water and 50% solute in the solvent.
Okay,
and
so
the other
that's
one,
another ISO. Yep.
And the other one is 75% water, 25% solute
Mm-hmm.
in the cell, and 80% water and 20% solute outside the cell.
Okay, cool. So you've got another example there of isotonic, and you've got one slightly. Sorry, I can't remember those figures that you just gave me. Can you tell me the second one again?
Hypotonic. So 75% water and 25% solute inside.
Yep.
And 80% water and 20% solute outside.
Okay, so slightly hypo. Yeah. Okay. So basically, and this obviously when we get to the next sort of application, slightly hyper, slightly hypo. Obviously, they're still going to have the effect. So osmosis is always going to work against the concentration gradient. It's always going to try and dilute. But if it's only a small difference, that the movement is going to be a lot smaller, obviously. Yeah, the more osmosis, the volume and the rate of osmosis will be faster with a bigger difference always, which is actually one of the reasons why we can correct a lot of body fluid problems, clinically speaking, because even if we have a situation where somebody is hypertonic, where there's way too much solute in their blood, it depends on the percentages, but as long as it's not too big, we can reverse it. Right? If it's really big, so remember, total cell crenation and total cell lysis are big events, right? They're right. That's at the end of a really, really bad imbalance between cellular fluid and what's going on outside. Most of the time, what you'll get is, again, a little bit hypertonic or a little bit hypotonic, and what you end up with is cells that aren't real happy, but they're not dead. Does that make sense? So if it's only a small amount, the cell's only affected a small amount. So it reduces function and it makes things, just not happy. But at the same time, it's not actually going to kill you. Whereas, of course, cells bursting does kill you, absolutely, as does crenation on the other end. All right. So that's our egg experiment, which I want to do with you live, quite frankly, because it's just much more fun than actually just thinking about it. Anywho, we can't. All righty. So this is having a look at your, and this is only for interest, by the way. You're not expected to remember this at all. I just thought it was interesting. So this is your different concentrations and where we give them. You probably aren't up to this yet, but you will be eventually. So what we give and where we give it. Okay. And it basically gives you some ideas of concentration there. Okay, so for instance, your intracellular and interstitial are often interchangeable. Sodium chloride and glucose, intracellular and interstitial. Colloids going into the plasma volume. Balanced crystalloids going into interstitial and lymphatic. Too much information, but point is, lots of different concentrations, but the point that we're trying to make here is always keeping everything isotonic, always, depending on the environment that it's in. Okay, but you don't need to know that chart. Please don't at this point. Okay, so I just really answered this for you completely. Someone else can answer it in their own words. What would happen if we delivered, so poor old Mr. Smith comes in and we deliver a hypertonic solution. What have we done to Mr. Smith?
Dehydrate him?
Dehydrate.
Yeah, that's right. He's gone like a prune. But more importantly, his cells have gone like a prune and crenated, which means that they're not working anymore, which means we're probably going to kill him. What happened?
We killed him.
We killed him. That's right. That wasn't a very nice thing to do. It's not considered good for a nurse to do that. And what if we gave him, we said, oh, he's really dehydrated. Let's just give him some distilled water into his veins.
You can't just put water straight into the veins.
No, you can't, because it is hypotonic. So it's going to make his cells explode, which is also generally frowned upon in the wards, I believe. So try not to kill people. That would be good. So that makes sense, everyone. And I'm not going to tell you that this is definitely going to be in your next test, but they do love to ask questions about hyper, and isotonic solutions. It's a big one, mainly because it is such a big one for your profession. Like the whole IV thing is just part of what you do, and knowing it is really, really important. Yeah. Okay. How are we going? Is that it? Have you guys got any more questions, or is that it?
That must be it.
Oh my God, we just like screamed through that. The good news I think about that, as far as I can see, is that you guys all seem to be pretty on top of most of that information. So is there anyone that is was absolutely at sea and lost through that entire thing? You know what I mean? You don't have to admit it, obviously. If you are, so let me just speak to all the people that maybe don't want to put up their hand and say me. If you are lost, that's Bronwyn did it. You brave girl. Okay, the answer to this, of course, is that you then need to go back and make sure that you can address all those learning objectives. Yes, it just means more study. That's all. You never give up. You just always go, well, apparently this is a bit of a weakness. I'm going back. You got to make that your slave. When will you get the results for the mini test? I would imagine within this week. So generally there's about a 3 or 4 day turnover for marking. So it should be pretty shortly, and hopefully you guys are pleased. Yeah.
I'm sorry, I do have a question.
Yeah.
What was the word for cells exploding, like the proper word?
Lysis.
Lysis, thank you.
Yeah, Lysis, or in a singular, that's plural, lies, Lyse. 11th of April for the results on the mini test. It'll be before that, Rebecca. We try to mark things as fast as we can, because we remember what it's like to be students waiting on results. Right, and you're literally checking Grade Centre every single day going, did I pass, did I pass? But don't worry, even if you didn't do particularly well on test 1, take all the lessons that you got from test 1 and apply them to test 2. So as we said, if the short answers took you a long time, maybe rearrange the way that you're doing the test. Maybe you do some of the hard ones first, or at least you keep a really close eye on the clock. If you've been more than 10 minutes on those multiple choices, get out of there. I know you think you're going to die, Beck, but you're not. It just feels like it. That's what uni feels like all the way through. Lots of small deaths until you get to graduation.
Maybe a really obvious question. When are we looking at test 2?
I didn't actually look up your subject outline, but that test 2 is on this topic and the next.
Okay, I just couldn't see a date, and then I started having like mini heart palpitations that I missed something.
Yeah, well,
And just
it'll
oh my
it'll
God.
be at least not next week, but the week after, 29th of March. Oh, there you go. Yep. Cool. Thanks, Portia. Excellent.
Right.
Yeah, so you've got time, but please, every single week, guys, just get those learning objectives. This should be part of your studying. This is what you do. Get the learning objectives, write them down, highlight the ones that you can't answer, and go get that. Go find the answer, go learn it. Do not leave any of those to chance. Okay, because any of those, yeah.
Last in the first assessment, there was a GUI chat or something. Was one of the questions asked about a topic that was discussed in the chat.
Right.
Yeah. Are there going to be any for this one? Just to double check, because I really missed that one.
No, that question was about Chat GPT, not about chat within the topic.
Chat GPT, yeah.
Yeah.
Mhm.
So all that was saying was this is what the answer that Chat GPT spat out. So can you come up with an answer that is different from what Chat GPT spat out? Yeah, it's still asking you for the same knowledge.
Yeah, I haven't actually, because this is my first semester here, I haven't actually touched on Chat GPT, so I actually missed out on that question.
No,
to be
that's
honest.
okay. So what Chat GPT is, it's good to know too, Nas. Thanks for letting me know. So Chat GPT is an open artificial intelligence application where essentially you can ask it any question and it will answer it for you. It pulls all its data from the internet, so from scientific papers, from blogs, from everything, absolutely anything. And essentially it's a brilliant cheat tool, right, for students now. It's I don't know if you guys have caught up with it, but it's a genius cheat tool. So you can literally type in absolutely anything and it will spit out not just an answer, but a beautifully constructed scientific answer as well. Right? It's particularly good at science speak, very, very good at science, not so good at poetry and stuff like that, but really, really good at science speak. So the reason that was in the test, I suspect, is just to make sure that you guys are aware that we are aware of ChatGPT, and we know that you can look up all of the answers to all of the questions on ChatGPT, but we also have to get above that and say, what can you tell me that ChatGPT didn't spit out? What do you remember? What details have you got from your lectures and your notes and your readings that this response did not give? Does that make sense?
I get you now. Yeah, I get you now.
The
Yeah,
ChatGPT question wanted to know what it missed.
Yeah, that went over my head. I was like, what is this?
Yeah, well, that's interesting actually, because that's just you just weren't familiar with that particular technology. And that's interesting.
You just introduced it to me, so.
You know what I think? Well, that's actually, we might have a look at that. I'll bring that up with Nat, because I think because we're so concerned with it as a university and as staff members. No, you go, Lillian. That's fine, darling. See ya. We just assume that all students would know about it too, and that's a really good. That's okay, Amy. All right, I'm going to bring that one up with Nat. Okay. Oh, cool. Amazing. All right. That's surprising, but also amazing. So that really did throw you guys off. All right. I will bring that up with Nat, and we will make sure that perhaps we can adjust things in the next test, although now you know what ChatGPT is. Play with it if you would, guys, as well. So if you just type ChatGPT, capital GPT, into Google, it'll come up with a number of different platforms that you can click on and try it. You can ask it all the questions maybe from the last test and see what it spits out for you. But remember that you also have to know what we've taught you in lectures, because you can't just use a ChatGPT answer for everything. And you have to put in your own words anyway, because even if you use ChatGPT, you're still plagiarizing an AI. And you're not allowed to do that either.
How do you catch people from doing that?
Well, there's a lot of different ways, but one of the big ones, like how do I tell, is the answer is too perfect, grammatically and spelling, and the knowledge is just too perfect. It's like, all right, that doesn't sound like a human, let alone a first-year human in a medical course. But secondly, most of the things we will actually put through Chat GPT before we mark them and see what it spits out. So it's not perfect, but we do also compare. And we'll ask the question a couple of different ways as well, just to see if anything comes up. The other problem with using Chat GPT to answer your questions, though, is that the likelihood of having exactly the same answer as somebody else is massive, right? And of course, that's a big flag for us, right? If everyone's got exactly the same wording, it's like, oh, yeah, statistically, the chances of that are impossible. So it's got to be reworded with your own understanding and your own words. Yeah, Amy, look, you can try it if you want. I think better in your profession, particularly, to rely on actually knowing what you're talking about. Just a thing. I think when your sister on duty actually asks you a question about medications or body systems and you have to go Google it to answer her, you're in a bit of a pickle there, really. You need to actually know what you're talking about. All right, guys, I will let you run off. Yep, I will see you next week and go through those learning objectives, okay? All right. No worries. See you soon, guys. You're welcome.
Thank you.
Thank you.
See ya.